there are 4 types of primary tissue. That is connective, epithelial, nervous, and muscle tissue. And in this video, I'd like to focus on muscle tissue. Now, muscle tissue has 3 types: smooth, cardiac, and skeletal. When we look at these tissues underneath the microscope, we can see very distinct differences. So let's do a little quick example of each one. If we're looking at cardiac tissue, what we're going to see is long cylindrical cells. But in these cells, there's going to be these kind of branches where we're seeing the cell kind of go into different areas. We might see it over here too. So they're not really just necessarily laying on top of each other. Another thing about cardiac tissue is that they have stripes, or what we're going to call striations, along the lines, or along the cell. So they look like this. Another thing you're going to see with cardiac tissue is a centrally placed nucleus. So they're just kind of hanging out right in there. You might see another one somewhere over here. You're also going to see what we call intercalated discs, and they're going to be darker lines that are going to be going on from time to time in the sample. All right. Skeletal also has long and cylindrical types of cells. They are also striated. A lot of times they almost have a stacked appearance. They look like they're just kind of in a row on top of each other. It's a little different than this branched appearance that can really look a little bit more spaced out. One of the other catches with skeletal tissue, though, is that its nucleus is on the side, or what we call a peripheral nuclei, or nucleus. You know, whether we're talking about lots of them or singular, right? So here is our peripheral nuclei. I'm going to throw a loop on here for you, though. Every now and then, you'll see one that looks like this. And you're like, wait a minute, that might be a centrally placed nucleus. Well, it's not. What it is it's on the side, but you're looking at it being on the back side of this sample, because it really just depends on where was it cut when we took the sample. And this just is on that back side, so it appears like it's going straight through it, but really it's still on the side. It's not right there in the middle of the cell. The last type is smooth. Now, smooth is a little bit different for 2 reasons. Number one, we don't have any striations. So they're somewhat long and cylindrical cells, but there's no striations. And the other thing is we do a lot of kind of pointed ends on these cells. They also have a centrally placed nucleus, like so. Now, they're pointed ends. If you have a pencil, and I'm going to draw a really horrible pencil here. So here's a pencil, right? And then at this end, you've got that piece of metal, and here's your eraser. And on this end, we've sharpening. And then it kind of comes to a point like so, and then there's the lead here at this end. So that's my really bad drawing of a pencil. We call this end tapered. And when we look at smooth muscle, both ends of our cell is going to be tapered. So they're tapered at both ends, giving it kind of that pointed look, like a sharpened pencil. And that is really different than any of the other 2 types. All right, so here is 2 questions you can use to help you kind of determine what type of muscle tissue am I looking at when I'm looking at a specimen underneath the microscope. So the first question you're going to ask yourself is, are there stripes or those striations? And it's a yes or no question. If the answer is yes, you've eliminated smooth as a possibility, and we're looking at one of these two types. If the answer is no, then you've actually eliminated both of these, and you're done. You know what your answer is. It's smooth muscle tissue. But if it was yes, and we know it's not smooth, so here we are, then we need to go to another question. And there's a whole series of these that we could ask. The easiest is probably, where is the nucleus? It's either in the center or it's on the side. And then that helps us kind of just go, all right, well, if it's in the center, it's cardiac. If it's on the side, it's skeletal. If you have a whole bunch of these kind of on the back side nuclei that sometimes almost look like they're in the center and you're really questioning it, you need to go to another question. And that second question is, are there branches or are there not branches? And I didn't use that first only because sometimes the branches are really difficult to see. And so they almost appear more of these stacked, like skeletal. But if you can see the branches, then you also know that we're back to cardiac and skeletal is the opposite. The other thing is, if you are having trouble with the nuclei, they're kind of really throwing you, and you're not seeing any branches, everything looks kind of nice and rowed and stacked, then you might be able to find these intercalated discs. They come off as really dark lines in the sample, and so they sometimes will stick out, and you can see them. And they happen, kind of on either side of a nucleus as you're going through a specimen. So you can really eventually look at the two and see some differences, and hopefully that will help you as you navigate your way through muscle tissue.